So hopefully can you see the presentation?
Yes, perfect.
Excellent. So I'm going to start by telling you a little bit about Work Package 7. So to give you a bit more introductions, we work alongside Mathieu Bottier, who is a researcher at the Royal Brompton Hospital in London, and Nazreen Ruman, who is working currently in paediatrics at Washington University, but is also affiliated to the Al-Quds University in Palestine. And we've been meeting since the start of this new BEAT-PCD funding round alongside Myrona and Amelia, who regularly interact with our discussions. And the whole remit of our work package is to improve PCD diagnosis. So our objectives are quite clear. We want to improve the accuracy and accessibility of diagnostics across Europe through education and sharing resources. We've also taken a deeper interest into what digital tools are available to us and how people are harnessing the new wave of artificial intelligence to standardize and improve how we perform our diagnostics. But also this is potentially a strategy for non-specialist centres going forward. And we'd also, we're working towards standardization and unification of how we interpret our diagnostic test results. So if you bear those objectives in mind, the next few slides are hopefully to demonstrate ways in which we're trying to achieve this. So to start off with, I want to highlight that the registration for our next BEAT-PCD in-person meeting in Amsterdam is now open for registration. And this is to highlight that this is one of our key educational platforms to come together to share expertise between researchers, clinicians, healthcare professionals, also patient support groups, but also to collaborate with our ERN lung colleagues. So pay attention to that and go onto the BEAT-PCD website if you'd like to register. And another highlight of our year is the ERS Congress, which bolts onto the back of the BEAT-PCD meeting this year in Amsterdam, and we have an excellent representation of PCD at this year's meeting. So to give you some highlights, we've got poster sessions across most of the days, but I particularly want to draw your attention to the guideline session, which I'll come on to in a moment, which is on the Monday morning. But we've got two excellent presenters giving oral presentations on Monday. At 11 o'clock, Janice Coles from Southampton, my colleagues, talking about variants in dynein assembly factor genes. And we also have Mafalda talking about advancing PCD gene diagnostics. So we're going to be launching the new ERS ATS guidelines in Amsterdam. So to draw your attention to the Monday morning session, we have a section that Amelia is presenting. And this basically is to highlight where we've come from the original guidelines that were published in 2017 by Professor Lucas and Adam Shapiro in the ATS. There's so much convergence between our diagnostic methods, and this guideline will really underpin how, going forward, we should be aligning. I've jumped too many screens. There we go. So talking to how diagnosticians on the ground can actually interact with each other and engage with other centres for expertise sharing and knowledge transfer. We have a Slack platform that we instigated at the inception of our work package. It's basically a peer support platform that's available for you to download onto your phone or PC, and you can upload messages, images, and talk to experts in this platform that will basically signpost to other groups doing similar work or help to share knowledge on what information is already published. And we curate this at least once a week, and we have notifications come through. So everybody is welcome to feed into this. And if you look on the left-hand side, you can see the different channels. We've got general questions, we have genetics questions, high-speed video, immunofluorescence, and TEM. It is a free platform, so every 90 days we lose content. So sometimes you'll see not much on there, depending on how much footfall we've had. But essentially, you'll get quite a quick response to your questions. And this is something I would encourage you all to engage with, given that we have potentially over 80 members on this platform. So speaking to a little bit about what Panagiotis has led, maybe perhaps I should have let Panagiotis talk to this, but I'm doing the introduction section. So Panagiotis took on from the ERS task force that developed the nasal nitric oxide technical standard for testing in children. I want to highlight that not only is this technical standard being produced for specialists, but there's also been a Breathe article developed for non-specialist readers. So the component that we've contributed within our work package is a patient information sheet, and this is to help reduce anxiety in patients, carers, and even the patients by being able to understand what the test is for and why it is important. And in order to not only have this presented in English, developed with colleagues at Southampton University, particularly Amanda Harris, and the PCD Support UK, we've actually reached out to other members in BEAT-PCD who have enabled us to translate into over 10 different languages. And as these new leaflets are being produced, they will be uploaded sequentially to the BEAT-PCD platform for you to download and use in your clinics. So as we know, we have excellent consensus for transmission electron microscopy to diagnose PCD. And this is something that we advocate using across all diagnostic centres. We don't, however, have the same level of standard for high-speed video, which is something that isn't actually used across all centres. But as we can see from well, I'm sure it will be re-outlined in the new guideline that's to be launched, but we have evidence that if you have multiple occasions of high-speed video data showing an abnormality, there is a potential to label a patient as being PCD highly likely, which is quite supportive clinically. So as you look at the video, this is of a normal ciliary function. But how on earth would you start to describe this in words? Because it is quite a subjective analysis. So we're hoping that convergence of language that we've been able to develop in a previous BEAT-PCD questionnaire, we're hoping to develop training content to provide online training for those that want to take up high-speed video microscopy. And this will talk to, the different chapters will talk to standards of methodology, also expectations for sample condition, and also typically what quality of ciliated edge you should be assessing, and examples of dyskinesia, both secondary and primary. So hopefully this will be something that we can deliver for you going forward. I'd like to now cross over to Panagiotis. So I'll probably move the slides along and Panagiotis will now talk on a systematic review that we have been endeavouring to look at what literature is out there to look at use of digital automated tools and AI use in PCD diagnostics. So over to you, Panagiotis.
Thanks, Claire. Just to say that the team working on the systematic review is the people that are involved in the work package for the diagnostics in BEAT-PCD. That is me, Claire, Nisreen, and Matthew. We have involved 2 medical students as junior scientists working the systematic review, Mia Shanks from Southampton and Andreas Prodopapas from Cyprus. And we have contact expert Amelia Schumark with us in the team and methodological expert Mirofora Goutaki. And as Claire said, our aim was to really look into the literature and see where and how AI and digital applications have been used in PCD focusing specifically on diagnostics and disease characterization, and of course to see the gaps and see how we can move forward. In the last part, see how we can move forward. We are going a step forward from the systematic review. We are involving the Connect CRC of ERS, which is a clinical research collaboration focusing on digital health and how we can standardize digital health in respiratory care. And they will be involved at the last stage of the systematic review to, based on our findings, and discuss them, how we can move forward in PCD with the digital applications and the AI-based tools. Next slide, please, Claire. Yeah. As any systematic review, we follow the quality criteria that are defined in the different guidelines like PRISMA. We have looked 20 years back. We have searching widely used and valid databases like PubMed and BASE, and we also screened gray literature through preprint databases like bioRxiv. We have 4 independent reviewers and at least 2 reviewers screened each record, and disagreements were resolved by more experienced reviewers. We did the quality control assessment. The review was registered timely in a public repository. And the most important information, the inclusion criteria, we focused on studies that combined PCD or motile cilia and AI or digital tools, and aim to address screening of the disease, diagnosis of the disease, improving phenotype characterization, carrying out phenotype and genotype correlations, and disease severity. And we did not include studies that, of course, did not use digital tools or did not use AI. We did not include studies that focused only on CBF, cilia repeat frequency assessment. There were some studies like that, but we know now, after all these years of research, that cilia repeat frequency alone is not a diagnostic feature of the disease. So addressing AI tools only for that was not something that we are interested. And of course, because of the scarcity of data and for practical reasons, because in other diseases this has been done more extensively, we did not focus on studies that use AI to assess disease prognosis. Overall, we started from more than 700 records, and through screening, we ended up in 28 records to analyze in our systematic review. Claire, if you can go ahead. Of these, most of them were less than 50 patients. So these were mostly single center studies. Most of them, they were focusing on the diagnosis, digital imaging processing, and machine learning techniques combined with imaging processing. They address aspects like cilia repeating, particle flow, waveform, rotational polarity, and for high speed. And some of them focus on automating EM analysis and automating the IF analysis. A second large group of studies were those that were not involved in the diagnosis, but they were involved in carrying out PCD genotype phenotype correlations. There were also some more studies focusing on the biology, on the structural biology of the cilia. And there were studies that aim to address PCD symptoms and addressing clinical characterization through improving CT imaging or automating CT imaging. For the first theme, I think Claire will be the one doing the talking. So Claire, back to you.
So for theme 1, we looked at how automated or digital tools and AI prediction tools were used to enhance ciliary function analysis or ultrastructural analysis. And we found 17 studies that tended to use digital image processing, and they implemented aspects of machine learning into procedures. So most of them were based on how to interpret high-speed video microscopy, but I think some of the strongest evidence was supplied within the TEM imaging methods. And the N=1 is a study that we did at Southampton that I'll share with you at the end of this theme section. So I want to start by drawing your attention to the PAPON study in 2012. It was really the first time that anyone had taken high-speed video analysis and tried to strip out the subjective component, and they tried to develop ways to interrogate ciliary beating in a more quantitative manner. So this was a pilot study. It was highly successful, but unfortunately the software is only really unique to that centre, but it technically shows us what could be done if we were to develop our high-speed video methodology beyond our subjective analysis. So not only did they look at beat frequency, which Panagiotis has already said has got low sensitivity for finding PCD unless your cilia are always static, which isn't always the case. They looked at things like stroke duration, the pause after the forward and recovery stroke. They looked at ciliary length, they looked at the angle of beating, but also they looked at the distance travelled for the ciliary tip. So they had to basically have perfect side profiles of ciliary beating where they quantified the start point and the end point of the range of movement of the cilia. And they had to weight this value based on the percentage of beating ciliated edges. But they found that the distance travelled by the cilia tip weighted by the percentage of beating ciliated edges to achieve at least 96% specificity. So this graph here, which I chose the one from the paper that is set against the ciliary dyskinesia score. So this is basically showing any of the dots in black which were considered less likely to have PCD, and any of the clear diamonds which were based on PCD patients. The higher your abnormal beating dyskinesia score, the distance travelled of the cilia tip was much reduced, and it segregated quite nicely, giving that high sensitivity and specificity. And it also, by implementing tests like this, they found that it removed subjectivity to the point where it reduced inter-observer variability to distinguish PCD from controls. So I think this was a really lovely study for high-speed video, promoting its value beyond that of a subjective nature. Taking it on a bit of a different approach, quite a few different studies have looked at beating waveform. This is something that I don't have any personal expertise in, but I know that Patrick Sears at Chapel Hill was one of the pioneers in developing ciliary dynamic studies. And similar to the previous study, but slightly different, he wanted to see if he could model what shape of ciliary beating would contribute to an effective power stroke and recovery stroke, and could you actually quantitate the waveform in itself. And I think he successfully achieved this. The problem was that it didn't then get applied into comparing against different PCD patients. So this is, again, another example of a powerful analysis tool that wasn't particularly taken forward. The study by Quinn et al. Actually attempted to develop a computational pipeline, and they used computer vision to decompose ciliary motion into quantitative elements, similar to that of the PAPON study. And they focused quite heavily on, so I'm just going to have to move Panagiotis to the left. They focused on rotational magnitude and also deformation magnitude, which were, when you go into the paper, it demonstrates these as different aspects of the waveform between normal and abnormal beating. And what they found was that they got quite a clear distinction between what they used as their abnormal controls to their normal controls. And they tried to, they used this in 2 independent data cohorts, and they found this computational pipeline to be over 90% effective at designating who had congenital heart disease, and heterotaxy from those diagnosed previously with PCD from healthy controls. So again, looking at waveform, Cioccolì used a quantitative method, and he particularly wanted to look at curvature of the waveform in order to distinguish specific variants that reduced that curvature of waveform. So he focused on patients with genetically confirmed hide-in or DNH11 variants. And you can see quite clearly here, and we do see it when we look down our microscopes, you can see very restricted movement with DNH11, with very reduced amplitude, very stiff beating, usually hyperkinetic. And the hide-in case also didn't have had a severely reduced amplitude in the cases that he used. And this is essentially a method of quantifying that rather than having a subjective comment for it. So he did a principal component analysis, and he demonstrated that you could get quite good segregation of PCD cases from healthy controls. And the little bar on the right-hand side is just basically breaking down beat amplitude and curvature and force. And the darker blue the box, the more important that attribute was. So looking at a paper produced by Piffrey et al. In 2013, they attempted to use soft computing analysis to attempt to diagnose primary ciliary dyskinesia from secondary dyskinesia using a suspension cell culture method. So they had PCD patients who were TEM diagnosed and considered confirmed, and then anyone that had an EM normal who was considered to be less likely to have PCD was designated as having secondary dyskinesia. They generated spheroids, and they used attributes of rotation and migration and the ability of the cilia to move debris. And they found a good correlation in PCD that they had loss of rotation, loss of migration, and loss of debris movement in the PCD cohort. And they used an artificial neural network-based model to predict patterns in these data variables. And they found, dependent on these different attributes that they apportioned for rotation, migration, and so on, they found these to be high. The model was basically able to be quite sensitive and specific at determining whether it was PCD or what they considered to be secondary dyskinesia. I think the only caveat to this study was that there could have been cases of PCD hidden amongst the secondary dyskinesia that we should always bear in mind, because PCD can never be discounted. So moving forwards, before I hand back to Panagiotis, I want to draw your attention to excellent TEM analysis, which uses image averaging. So we all use transmission electron microscopy as a staple to diagnose PCD. And the Brompton group have been highly successful at implementing artificial intelligence platforms to see if they could find patterns in computer ability to demonstrate abnormalities. So they took TEM images from over 21,000 cilia cross sections to train their AI. They basically trained the AI to determine what was a good image in good cross section versus an image that the computer should discount and not use to make its analysis. And then they tested the PCD aid alongside their ordinary diagnostic pathway in 158 cases. And they found that not only could the computational method that built a TEM average, and then it demonstrated where there was loss of density in the image, in the pixels. They found that not only did it agree with their MDT process, their diagnostic pathway, but it also demonstrated and highlighted specific examples where deficits in sort of central sheath staining. Sorry, this is a volume, not staining. When volume of TEM went missing, they could demonstrate that this was more likely to be attributed to hide-in cases, and they went on to develop PCD Detect as an offline software that could be used anywhere. And they found that their artificial intelligence had a sensitivity of 87% and a specificity of 88%, which was absolutely excellent. But not only that, the TEM image analysis took under 1 minute per patient, which is something that we can't compete with. And there was excellent agreement, over 75% agreement with the specialist MDT method. And this is the study that we did at Southampton. It was a student project study, which we employed a plugin for ImageJ to see if we could take our excellent EM images and we could develop a method to mask over the green area that was tubulin stained, representing cilia, and could we overlay the red target staining on top of the tubulin, and could we quantify a loss of staining in true PCD cases? So you create the mask using the tubulin, you overlay the target, and you look to see if it agrees or not. So what we did was we blinded our analysis person who had to go through this process manually, and we applied a threshold to enable the mask to effectively fit over the tubulin. And we found that depending on how we thresholded, we could actually gain 95% sensitivity agreement with the human consideration for the IF staining, with 84% specificity. So sensitivity is where it agrees with your finding of true diagnostic PCD, and specificity is where it agrees with your finding that you think the staining is normal. And if you group together the different images from different antibodies, different targets like DNA Li1 or H5, so that's for an inner dining arm target, an outer dining arm target, a radial spoke head protein, and a nexin link protein, which excellently stain along the ciliary axoneme, and you group them together, you could see that when that staining was absent, it was absolutely absent compared to the true positive group. So we think going forward, a computer algorithm like this has potential to be automated, but not only that, we've implemented use of a slide scanner to generate a higher throughput of our image acquisition. And with that, I will hand over to Panagiotis for theme 2.
Thanks. And the applications of AI in diagnostics are very detailed and promising, some of them. And I think when we started the systematic review, that was our main, driving factor. But there is literature beyond that, and we were, for some of them, surprised to find them in these articles and show the potential that there is with AI. So the second theme relates mostly on how we better characterize the disease. And one way is to inform genotype-phenotype correlations, but there are a lot of applications on going to the basic science and using AI to better understand the structure and the function of the motile cilium. And this can have implications for the for later diagnosis or even drug treatment and interventions. Also, there are approaches targeting primarily electronic health records that we can go and find PCD cases that are undiagnosed. And also there is a huge area of research that touches also PCD, of improving with AI or automating the medical imaging, and especially lung function, lung CTs. And this can also help with PCD disease characterization. So Claire, you can go to the next slide. The first subsection of this theme was how we characterize the axoneme, the structural biology, and how we inform genotype-phenotype correlations. There were two studies, one from O'Toole, one from Walton. The one from O'Toole is a bit older. It does use digital methods to better visualize the axoneme. However, the one by Walton that is more recent, and I can only say that it's a hallmark study. It goes in much more detail using AI to reveal the structure of the axoneme. And we also show an example, the one from Piferi et al. On how they used these approaches to inform genotype-phenotype correlations. Claire, we will go to, yeah. This one by Walton, they did magnificent work. It was published in Nature in 2023. They combined cryo-electron microscopy, which is probably, as some of you may know, it's a more advanced electron microscopy method that flash freezes the samples. And they combine that improved imaging with AI-enabled protein structure prediction. So we have improved imaging combined with prediction of protein structures, and this allowed them to create very detailed atomic level visuals and maps of the ciliary axoneme, both for Chlamydomonas and for human motile cilia. And for both species, they describe the structure, the periodicity, and the interconnectivity between different proteins. And their results, they show both the common features of the two species that shows the evolutionary conserved dynamics that we have, as this mechanism is shared in two very distinct species, but also the differences that highlight the elements that actually lead to the different waveform between the Chlamydomonas and the human cilia. And you can just see the examples of these images on how they have been able to visualize the INDAs, the ONDAs, the radial spokes, etc. You can see both a longitudinal section. This is one of the longest repeats, the 96 nanometers repeat. There are also lower periodicities, narrower periodicities, if I frame it better. And you can see the cross-section of the human doublet, of a doublet of a human cilium on the right side. If we can go to the next slide, yeah. And beyond the normal samples, they also used samples from patients with specific genetically confirmed variants of PCD. And they showed that with this method, you can better visualize and you can see effects on other proteins as well. So for example, in the lower panel here that we have abnormalities in the CDC3940 genes, we also see absence of specific proteins in the doublet, additional proteins. And this goes forward that you can use this technique to actually explain patients that have not been genetically confirmed and you are seeing additional effects, or you can, and the authors make this case very strongly that now that we know which proteins and how they interact in the axoneme, they make a case that clinical genetic testing can expand beyond those that are already confirmed as PCD genetic defects. So we know the proteins that are structurally there, we know how they interact, so we can expand our genetic testing, include these proteins as well, these genes of these proteins, and this can probably explain the 20 to 25% that we see abnormalities in TEM, we see abnormalities in high speed. We classify them as PCD patients, but their genetics, even in expanded panels of 50 or 60 genes, they are still not confirmed genetically. So they make the case that if we put these structural confirmed proteins now there, we may explain some of these missing genetic diagnoses. Claire, thank you. The other one in this subsection is the study by Piferi. They used a combination of principal component analysis followed by neural network algorithms to create clusters of structural and functional phenotypes, and they correlated the functional and structural phenotypes. These I'm basically talking about high-speed video microscopy characteristics and TEM findings with specific gene groups. This shows how this technique can be used for genotype-phenotype correlations. The study is not clear on how the groups that they were comparing with, actually they were structured, why these genes were grouped together and why the other group included those genes. But definitely shows the methodology, how this can be applied. It can be applied to single genes in the future, or it can be applied to, instead of having the phenotypes, the diagnostics, having the clinical severity or the lung function or other parameters that can be combined with the genetic defects. Next slide. The next one is one of the most interesting that I found in this theme, is how we can use data and big data to better screen for PCD. And there were three studies here. The first one by Rother was one on focusing on questionnaires. The other two were focusing on electronic health records, and we'll see an example of both. Next slide. The one by Rother, they did a study. They gave questionnaires to parents of children with different respiratory diseases. So it's not only for PCD. They gave it to parents of different children with different diseases, and they asked about their symptoms. And based on the answers of this questionnaire, they created a tool to distinguish between the different diseases, and they tested that in a new cohort of 170 parents of children that came with possible respiratory diagnosis, and they were able to see that our questionnaire with this tool based on AI was able to distinguish between these diseases. And for PCD, they were able to distinguish 100%. They were able to find the PCD cases through this tool. For the other diseases, they were a bit lower, but still this shows the promise of applying this tool even on a questionnaire-based approach. Next slide. And they also did heat maps of the different comparisons of questions between different diseases. And for example, between CF and PCD, the two questions that actually drove this distinction between the diseases was the ones about ER infections and ER symptoms. So they were able to distinguish between PCD and CF primarily based on these questions. The other one is a preprint, so it has not been published yet. It will definitely go under peer review, so it's maybe different at the end, some of its findings or its interpretations. But basically, we have an excellent study from the US that tested a random forest model in electronic health records to test for PCD. And it has been done in a huge number of subjects. So they created a model based on confirmed PCD patients from the North American Consortium, the PCD Foundation, the Patient Association of North America, combined with people that they have been tested and they have been classified as specific congenital malformation of the respiratory system. So not exactly PCD, but classified in the electronic health records as something very close to PCD. And they also had a TEM testing carried out. And based on this group of people, they identified the diagnostic, procedural, and pharmaceutical electronic health records codes that correspond to these patients, and they compare them to a random group, a large random group of people in these electronic health records that they did not have these characteristics, these PCD characteristics. And they were able to create a model to distinguish between the two. And on the left panel here, you see the positive cases. They were selected from the PCD foundation, and through this classification that I said, and the background class, this is the controls that they compared together, and they were able to test how well our model distinguishes between these two, and then they applied it to 1.3 million people in this database, in children in this database, and based on the estimated prevalence in the US, they expected to have about 8,000 people as PCD, and they tested whether this actually was picked up by their algorithm. Next slide. Great. So in terms of diagnostic accuracy, this algorithm was pretty good when it included also the genetic malformation of the respiratory system category. It had a sensitivity between 80 to 70, 80 to 90%, and a positive predictive value above 50%, which is pretty good for a rare disease. And when they applied it to the entire database of 1.3 million children, they picked up 7,700 PCD positive patients when they expected about 1,000 greater. However, this is the same order of magnitude, so they went pretty close. And the estimated prevalence that they expected was 1 to 159, and they got up to 1 to 179. So that's pretty good. That's pretty good. And they also were able to classify which characteristics, or better, which electronic health record codes actually predict the PCD case with more importance. And this one was sinus inversus, and some expected ones like nasal biopsy, but also airway clearance, lavage, and some other interesting findings. Next slide. Lastly, we have clinical characterization. We have a lot of automation of imaging studies here. Some of them were used to better describe the disease. Some of them were used to distinguish between PCD phenotypes with other diseases. But I will focus only to one as an example. If you can go ahead, Claire. The one by Huang, a French study that they used automated. I think it's the first one that used automated CT scores in PCD for automated lung CT scores for PCD. And they showed that when you do image analysis of the CT scans, you can estimate different densities for the lung, and there are different metrics that you can get for the lung CT, like mean lung density, the kurtosis, and the skewness. And based on this, you can objectively quantify lung disease in these patients. And they show that, for example, here that this density histogram, the black line shows milder disease compared to the gray line. And if we go to the next slide, they were able to first show that the automatic scoring was very well correlated with the visual scoring, but much faster and objective. And also that it was able to predict meaningfully lung function cross-sectionally. It was not validated in an independent group, and is not validated prospectively. So we don't know if changes through these scores, objective scores, mean changes in lung function or other disease characteristics, but it shows great promise. And I think we'll see more studies as we go forward with lung CTs and AI. And lastly, to conclude, so we know that AI-based tools and digital automation tools hold promise in improving and standardizing PCD diagnosis. And the good thing is that some of them are available already, some of them are open, and this can increase validation of these tools in different centers, because as we said, currently we are at the single center stage. The recent developments in protein folding, like AlphaFold and different softwares, they can help our ability to understand the axonomal organization, the protein interactions, and also characterize the pathologic variants. And very promising is the automated and reliable screening of electronic health records, even in the absence of a specific PCD-ICD code, and it can help us find and diagnose individuals. Also, with the AI-based, we can do better analysis of our CT scans, faster analysis of our CT scans, and this can especially help us detect earlier development of bronchiectasis, especially in PCD, where CT scans are not done very routinely. We have greater gaps in the years that we do CT. And to close, I would like to say, as Claire highlighted, there are a lot of posters and oral talks in the Amsterdam meeting of ERS. We will be presenting this work in the session that is full of PCD in Tuesday, 30th of September at noon. Back to you, Claire.
So this is just to highlight, because I have an opportunity to, we gained some grant funding in the UK to do transcriptomic analysis pipeline automation. So leading on from our systematic review, we're looking at ways in which AI and digital tools can be used, but we're trying to apply a novel approach to transcriptomic analysis in PCD genetics. So we've used the funding to appoint Dr. Carolina Jaramillo-Oquendo, who is working alongside our PhD student Lee Baker, and this is following on from work published already by Jelmer Legerbeck. The premise is that we take nasal brushings from our patients. They've been through our routine diagnostics. We use cell-cultured material to extract RNA and gain sequencing data. So it's bulk sequencing with a high read depth. The original pipeline was done sequentially and manually in a blinded fashion, but going forward, we want to now expedite how we come to our conclusions. So Lee Baker has been instrumental in finding ways to automate the pipeline using script to run the data in a faster manner. And the outputs, we are hoping that we can introduce machine learning algorithms to interrogate, looking for variant calling and abnormal splicing events and abnormal gene expression events. So the premise is that we'll have all available data on the patient going forward, clinic history, clinical characteristics, diagnostic attributes and outcomes of tests, but also signposting from blood genomics. Particularly, our inclusion criteria focuses on whether patients have already got a pathogenic or likely pathogenic variant, combined with a lack of second variants or perhaps even presence of a VUS of unknown pathogenicity. That's a variant of uncertain pathogenicity of VUS. And we want to be able to basically put this into a computational system that will drive down to a particular PCD gene of interest, but not only that, would be able to variant call and demonstrate expression abnormalities, and particularly find abnormal splicing variants that are generally underrepresented on genetic databases. And going forward, we also have another study that's led by Matteo. He's looking at implementing AI across the whole of PCD diagnostics at the Royal Brompton. So not only is he going to take forward the PCD aid methodology that I've demonstrated and cross-validate that with Southampton and the University of Leicester, he's also going to look at ways to look at optical flow and whether he can apply AI modeling to classify abnormal beat patterns. So this is driving on from the Papon paper where Matteo started his PhD in Paris. But he's also hoping to deliver a PCD aid-like model for immunofluorescence. And he hopes that this will be an easy to use platform that can be given to others to be able to expedite their own diagnostic testing methods, and also be something that increases the throughput of our patient sampling. And this is in collaboration with Computer Vision and Intel Getty. And with that, does anybody have any questions for us? And I'll stop sharing so I can see your faces.
Excellent talks, both of you. Thank you very much for this wonderful overview of how it all started with like clearer assessment and clearer imaging diagnostics up to atomic imaging, and then on that, based on that, developing those AI tools. Thank you both very much. And yes, the
Thank
audience is
you.
joining in and clapping virtually for you. Maybe while they type in their questions, let's kick off the Q&A. I have some questions for you. I think you showed very clearly how it was based on specific populations with the data that was then fed to the AI tool, and then that AI tool using that information to then help with diagnostics and PCD work, diagnosing, fishing out any undiagnosed PCD patients before. Do you have any knowledge on how those tools that were fed and based on specific populations, how they perform in other, maybe more genetic, diverse or a completely genetic and phenotypic different population?
I think if you don't mind me going first, Panyotis, yeah. So the we already know from the genetic study that looked at specific databases to look at the potentially true prevalence of PCD. And over the years it's come down, it's gone from one in, I don't know, lots of thousand down to 1 in 7,500 predicted. But if you look in different populations with different demographics, you get a different top 10 different variants coming out as your most prevalent gene. What the argument is by ethnicity, although arguably that needs to be looked at as well, because populations move around to the point where really you should be looking at areas rather than original ethnicity, perhaps. And I don't think any of these preliminary studies introducing AI have actually addressed any of that diversity, and a lot haven't been cross-validated. A lot have used what they argue to be expert panels to design what their questions are. And I think this is something that needs to be looked at less with less opinion. There needs to be better modeling of data available so that we have less of a say in what parameters are the most important. I think that preprint at the end highlights a good example of using as much available data, but even that study didn't take into account medical information available from, it's not Obamacare, basically they use private medical records. And I think that could be expanded into more diverse communities, even in across America, before they start looking, cross centers and in different countries. But I think from an epidemiological point of view, my colleague Bruna Rubo is working with BEAT-PCD members to look at what is important across different what outcomes are important and what lack of knowledge, what patient representation is requiring to get PCD diagnosed better by demographic region. I think the bigger that we can think as a community, the healthier these analyses will be. I know it's a very general answer. I think the bottom line is no one's done this across different centres.
If I may add, it's correct that it's not done in different centers, and I think when it's going to be done in different centers, it cannot be done in the same way. You cannot put a model from one system, health system, to another one. The populations are different, the practices are different, even the recording of whatever records we may apply them to will be different. So I think the principle needs to be evolved, and the principle needs to be repeatedly tested in the other systems, and probably variations of the same principle will be more effective in different systems. So I think this is how we can address the variability in the populations and the different systems that we have.
I think the CRC Connect focus group that we're going to add on some into the systematic review will be quite insightful, because this will bring together experts in the field that are looking at how digital tools and AI will be implemented into our diagnostic and clinical processes, based on their expert opinion, with a lighter touch of PCD perspective. So this will hopefully give us some insight to bring to the systematic review as we deliver it.
Great. Thank you very much. Yes, I think that would be probably the next 2 or 3 steps into the future. And also probably maybe, just as you said, not just to retest in the same population, or maybe then also just connect with those, not just the databases, but also then on those based on AI tools, right? So these are very exciting times we can look forward to. And Mina, do you have a question?
No, that's very, very inspiring talk. Thank you. Great talk. I have no question. AI has lots of unknown things. So
I think the bottom line for us in the work package is we're diagnosticians, cell biologists, epidemiologists, clinicians. We're not AI experts. We do have bioinformatics colleagues. We've got people who do coding. But the bottom line is we wanted to bring together evidence to put into the community, that not only can you read this, it might signpost to interesting applications and digital tools, but we're not experts ourselves. This is something that we're trying to navigate together as a community, and I think that's the important message. It's quite a difficult subject matter, and I guess we're here today to tell you what we've learned from our perspective. But going forward, it will change over time, and I think a presentation in a couple of years' time will be telling you a different, the advancements and what it's really doing, and the pitfalls and the problems that lead from it. A lot of these AI trained systems, you can introduce errors, and it has to be carefully managed. You can do supervised training of systems or unsupervised training of systems, and I think the bottom line is that learning how to navigate the strengths and weaknesses of different methods, and having bioinformaticians that really understand the processes to guide diagnosticians and researchers and clinicians like us is going to be really important going forward.
Absolutely. Yeah. Very, very broad and multidisciplinary research in that very rare, broad disease. Maybe let's give our audience some moment also to think about what questions. Meanwhile, I'd like to ask another question. And do you have any information on bias assessment of the performance of AI, how they performed in stratified by age of patients, or the method of how the cells, or whether those were nasal brushings or by bronchoscopy, or how old those diagnostic information were, how that, if there was any bias assessment, or if there are plans to do that.
I don't think there was enough evidence provided in some of the papers to give you that confidence. And I do think that even as we were assessing the data on some of the papers, our own considerations were that there was probably quite a lot of bias going into some of the groupings of how systems were trained. So I think for me, the best example is the preprint, where they've tried to go out and look at electronic records with less bias, and they've actually looked at the variables and seen which ones create more weighting into their AI output. So they've been able to categorize which variables are more important rather than having a selection panel that's chosen what they think are variables of higher importance. So I think there's probably quite a lot of bias. And I guess going into these papers, which hopefully our paper will signpost you to and give you some detail on, I think as you go into them, you'll realise that there's probably quite unsatisfactory evidence of how bias was reduced in some cases. Do you want to add anything, Panagiotis?
I just wanted to add that it was characteristic when we did the bias assessment, the quality of bias assessment on these papers. Based on the parameters that we're looking for, in general, they are okay. However, for the specific parameter, whether they were controlling for confounders and whether they're doing more rigid analysis in terms of statistics, most of them, they were low. So it shows that, as Claire said, that there was we were missing some confidence in some of these papers. So there is room for improvement for sure.
Great. Thank you both again. Very clear and direct answers. Thank you very much.
Oh, we've got something in chat, perhaps?
I don't see any questions right now. Maybe they were just flabbergasted by your expertise.
It's the AI assistant helping me to take notes.
Yes.
Oh, lovely. If anyone has any questions they'd like to direct to us.
Yes, we
Sorry,
are two people
Panagiotis.
now.
Yeah, that's the most creepy part, the one that comes and takes notes.
Exactly. AI everywhere.
Yes.
Yes, Zeynep says, thank you very much for this insightful online session. And also Ella said, thanks for this inspiring topic and talk.
I think everything is clear. So nobody has question. Thank you both of you.
And
you're great.
You're welcome to email. Welcome to email us with anything specific, and we'll see you at BEAT-PCD and hopefully the ERS.
Absolutely wonderful. That was a great ending of the talk. Thank you both very, very much. Very clearly wonderful talk. You're both wonderful speakers, as we already know, per usual.
Thank you.
Thank you very much.
Thank you very much.
Bye, everybody.
Have a nice evening. Bye.
Bye. Have a nice evening.